Okay, well, I think we've got a good number of participants to get started. I'm sure there will be some more trickling in as we go along, but I don't want to keep our speakers waiting and keep our participants waiting who joined on time. So welcome everyone to the first of our OBON webinar series, in which we'll be showcasing new and existing OBON endorsed projects. And these have been grouped by themes, so we'll be covering a different theme in each of the webinars, with today's kicking off on the topic of reference libraries and projects that are focusing on reference libraries. I would like to start with a brief overview of OBON itself, for those who are new to OBON or who haven't been involved for very long. Nick, was that a deliberate hand or no? Okay, no problem. Right. So my slides aren't moving. Okay, so for more background on OBON, this is going to be a brief presentation, but you can find out more by reading our strategy, which was published earlier this year, and it's available at the address provided here, and you can also use the QR code. So in a nutshell, OBON's vision is to accelerate informed decision-making to restore the health of our ocean using the universal signatures of life on Earth, biomolecules. And our mission is to create a hub for biomolecular measurement of marine life. We have 4 objectives which guide all of OBON's activities. The first one is to build a coastal to open ocean multi-omics biodiversity observing system, in particular by developing autonomous sampling and analysis capability, as well as promoting methods and technique development. So this is a very big goal, as you can see. The second one is to enhance marine omics data systems and ecosystem models by adding biomolecular components. Again, quite an ambitious goal, but this is a decade-long program, and probably will extend beyond the duration of the ocean decade. We also aim to partner with marine conservation and management specialists, policymakers, the private sector, and to address sustainable development. And another major goal and objective is to develop and transfer capacity through training programs, combined with funded equipment programs supported by development aid agencies and philanthropy. So this diagram kind of illustrates the OBON strategy and how it's articulated around these 4 areas, which are kind of aligned with the 4 objectives that I presented previously. So around developing networks and resources, innovating and developing the technology required, enhancing the use of information from biomolecular observations, and ultimately informing policy and a sustainable future. So OBON is a UN Ocean Decade program, and underneath Ocean Decade programs sit a number of projects. So under OBON, we now have 20 projects, and these are the projects that are being presented in this webinar series. We've started gathering information about the sampling locations that OBON and OBON projects cover. So we're starting to get quite a good global coverage, although we do have mostly coastal sampling stations. In each of the calls for new project proposals, we do highlight that we are very much open to endorsing open ocean projects, and also to collaborating with programs like one-off research cruises. So they may not be sampling repeatedly at the same locations, but even if it's just a one-off cruise, we're very happy to work with you. And now there's a mechanism through the Ocean Decade which allows that by proposing contributions rather than projects. So in terms of how we're organised, we have a coordination office, although this function is currently being filled by the POGO Secretariat. I completely forgot to introduce myself, but I'm Sophie CA. I'm the CEO of POGO. And as I showed on the first slide, POGO is the Partnership for Observation of the Global Ocean, and it's the lead organisation for OBON. And we have Luz Rodriguez, who's working as the OBON program manager. So sort of between the POGO Secretariat and Luz, we're covering the function of Program Coordination Office for OBON. We have a scientific advisory committee, which is very much international, which guides the OBON's activities and the delivery of our strategy. And we're in the process of setting up a user advisory committee, and also setting up various working groups that are currently in sort of different stages. And I think this is my second to last slide, just to highlight some of the activities that we've already undertaken or that we have in the planning. We had earlier this year an OBON workshop at the Westpac conference in Bangkok. We're planning a workshop on eDNA for fisheries management for next year, early next year, and also a workshop for the development of an eDNA network in Latin America and capacity development in the region. So just finally, the reminder of the OBON website. Margaret Leinen is the chair of OBON. She sends her apologies for today's webinar, but she's also happy to receive direct communications regarding OBON. Okay, so in terms of the format for this webinar, we have, as I mentioned previously, we have a number of projects that are working on reference libraries. We actually have 4 endorsed projects that are that were invited to present in this webinar. Unfortunately, one of them was not able to attend or to provide a pre-recorded presentation, which was the National Biodiversity DNA Library project based in Australia, led by Jenny Giles. But we do have 3 speakers with us today, representing 3 different projects. So in order of today's presentations, we'll have, first of all, Hugues, Roest, Crollius from the Institut de Biologie de l'ENS, representing the ATLASea, Atlas of Marine Genomes project, which is based out of France. Then we'll have Nicolas Pade, representing EMOBON and the eDNA Aquaplan project, which is both of those are European projects. And Nick is the director of the European Marine Biological Resource Centre, EMBRC. And finally, we'll be moving over to Canada, to the Marine Biodiversity Library and MPA eDNA monitoring project, which will be presented by Kristen Westfall. So we'll have each of the presentations followed by a couple of minutes for direct questions to the presenter, and then we'll have some general discussion for the last 30 to 40 minutes of this session, and have kind of a bit more focused discussion on what these projects could do together under the OBON framework. Okay, so I would like to invite Hugues to, if you could please share your screen, and then get started with your presentation. Do I need to unshare?
No, I think we're good.
No,
Thank you very much, Sophie, for the introduction. Can you hear me well?
Yes.
Great. Okay, so thank you for the opportunity to present our project. As Sophie mentioned, we just joined OBON. We were endorsed earlier this year, and as you might see, we are not a typical action within the OBON program, because we're not really concerned with observation as such. But I hope by the end of my presentation you will see why it makes sense that we joined OBON, and how complementary we can be to existing actions. So ATLASea is a project run by or funded by the French recovery plan after the COVID epidemics, which funded a number of projects in research in France. And it's run by the CNRS, the French National Research Agency, and the CEA, which is another French research agency, which in fact manages the French Sequencing Center, the main French Sequencing Center. So ATLASea is, I'll go into the objectives in a minute, but just to give you an outline, ATLASea is interested in sequencing marine organisms in the French economic exclusive zone, about 4,500 reference genomes. So we're aiming at sequencing 1 genome per species for 4,500 species, coming each from one individual sampled in the wild. And to achieve this, we've organized a project according to this diagram in 3 sub-projects, which are the 3 purple shapes in the middle: DiveSea, SecSea, and BiteSea. So DiveSea, as the name suggests, is the sampling sub-project of ATLASea. You see here in red the 2 main pilots of the ATLASea program, and in blue the other main partners, which are the Museum of the Musée National d'Histoire Naturelle, the French Natural History Museum, 3 universities: Sorbonne Université, Paris Sciences Lettres, and Aix-Marseille University, and Ifremer, that most of you must know, which is the French agency concerned with everything to do with the sea. So DiveSea is run by the Natural History Museum, and samples specimen directly along in the French EEZ with Ifremer and CNRS. Then samples are brought to Genoscope, which runs SecSea, so it extracts DNA and sequences and assembles the genomes, and then annotates the genome as well to find the genes. And BiteSea is responsible for the informatics infrastructure, collecting all the metadata, analyzing the genomes, and presenting the genomes into a dedicated web portal. And we have a number, so the sequences are going to be used, of course, by the research community and will be freely available. But we will fund 2 pilot projects that will come, I will describe in more details in a minute. Just to give you a little overview of the way the project is organized. So they are the 7 we call founding members, which I just mentioned. We have a number of research infrastructure that work with ATLASea. So the French Bioinformatics Institute, the French Genomics network of genomics platform, EMBRC France, which is working alongside the French marine stations to collect specimen. ERGA, the European Reference Genome Atlas project, which aims at sequencing like ATLASea, reference genomes for European eukaryote species, and Tara Ocean, which, of course, as you know, travels the seas and to sample to sample planktonic specimen for metagenomics. So the project is directed by myself and Patrick Vinker, who's also heading Genoscope, and we have a little operational committee that meets every week, which has the coordinators of the three projects, DiveSea, SexSea, and BiteSea, and our project manager, Camille Safransky, who will join the OBON meeting in Plymouth and who's attending this webinar. So just to give you an idea of the international context in which ATLASea is set, there are a number of projects that are interested in sequencing biodiversity at large, either across geographic sections on the planet or across taxonomic sections. So, for example, for two geographic-centered projects, you know of Darwin Tree of Life, probably, which is interested in sequencing all eukaryote species on the British Isles and Ireland. ERGA, which is interested in sequencing European species on continental Europe. And ATLASea is part of the ERGA project. And all these projects are part of the Earth Biogenome Project, which is, again, here an umbrella organization trying to coordinate all these projects who aim at sequencing reference genomes for biodiversity on Earth. Other projects are interested in specifically sequencing, say, vertebrates or fish or certain plants across the globe. Another regional project is the Catalan Biogenome Project in Spain. There's the ELIXIR European infrastructure that coordinates bioinformatic infrastructure, with whom we're also working, and of course the United Nations Decade of the Ocean through OBON. So more precisely, the objective is to sequence 4,500 species in the French EEZ, mainly in hexagonal France, as we say, metropolitan France, but also in overseas territories. These will be frozen, at least the tissue will be frozen, and vouchers will also be frozen in the Natural History Museum. And we will provide all the data freely to the community for various analyses, together with various analysis tools as well. We have a strong training program in ATLASea with dedicated summer and winter schools, funding for PhD grants and master bursaries. We also have a innovation component, so to say, where we will be funding public-private partnerships, so academic lab and a private lab. We will fund the academic lab through small grants to spearhead some projects linked to the use of genomic information for innovative programs. And also we will work with specific consortia that are yet to be created to work on two pilot projects, which I will describe in a minute. So the number of species we're targeting comes from this list elaborated by the French Biodiversity Office that every year updates a list of species that are known to exist in the French territories. And in the marine space, there's about 12,000 species that are known to exist in mainland France. And so this was our kind of reference to say we will sequence a large fraction of those. And although initially we were going to target specific species of high scientific interest or economic interest or endangered or emblematic or species with various criteria, we're now going for a kind of all-out effort to sample everything we can find that is not yet sequenced in specific areas, and sequencing all of those, because sampling is appearing to be a serious challenge these days to find suitable species for whole genome sequencing. So the first project is DiveSea, the first sub-project of ATLASea, and we have a number of sampling missions planned for this 8 years of the project. We're now in year 1 of the project. So they're mainly located at marine stations along the French coastline, but also deep sea campaigns by EMBRC and also some overseas territory. Here are some of the overseas territories which we're aiming in the Indian Ocean, in the Caribbean, in the Pacific Ocean as well. And more precisely, this is a calendar. We had a little pilot expedition in the Mediterranean in Le Cat like a year ago. We were in New Caledonia in March, on the Brittany, coast of Brittany, at Dinard, the marine station, in June and July. And just at the moment, so literally tomorrow, there is an expedition going to the Caribbean in Guadeloupe, and there is an ongoing expedition in the English Channel with EMBRC on a big boat of the French oceanographic fleet. So these are more expeditions planned for next year, and the following ones are not yet in the calendar, but there are more to come. Every June, we should visit, in blue here, a different marine station in France. So just briefly, for the sequencing, as I said, we're talking about reference genomes, so the complete genome assembled in chromosomes. This is done different sequencing techniques that are going to be used, as illustrated here in color. I won't go into the details, but I'd be happy to answer questions if you want. But basically, the idea is to do a first survey of genome size, genome heterozygosity, polyploidy, and so on. Then choose the best long read technology to
Hugues, are you still there?
Yes.
Okay, we lost you for a minute or so, but you're back, so please carry on.
Did you shall I go? So how long did you see this slide, or?
Yes, yeah, well, I think we got most of that slide.
Okay, so just to focus on the 2 pilot projects which we're going to launch starting next year. One is interested in studying the metabolic pathways leading to molecules of interest from marine genomes. And the other one is to study ecosystems subject to invasions. And so this project will be funded through calls for letter of intent, and based on successful applicants, we will form a consortium for each project that will be funded to run the project. Now, just a few words on the missions, on the sampling missions, which we have already run. One, as I said, in the Mediterranean last year, and 2 this year in the Pacific Ocean and in the coast of Brittany. So in Le Cat here, where the red arrow is, this was really a pilot project to start establishing our standard operating procedures on exactly what it means to sample from the wild, bring it in good quality to the lab, sequence or extract DNA and sequence these genomes. So these are a few samples which we found in the Mediterranean over there, and we sampled approximately 100 species, which have mostly now gone through the process of being having their DNA extracted, and they are being sequenced at the moment. In March, we went to New Caledonia here, just on the east coast of Australia, where we had assembled this team of divers and a large boat that could also sample offshore. And we sampled about 170 species, most of them, so about a third of them new to science, where we could assign the genus but not the species. And those unfortunately took a long time to come back because of troubles that we had there, where the airport was closed. So they only arrived in France last week. although the expedition was in March. And then this summer we were in Dinard. This is the Brittany coastline, just here. And Saint-Malo, for those who know, is on this side. Dinard is on this side here. And the spots here, the colored spots, are where we sampled fish using nets here, or divers where the colored dots are. We sampled approximately 460 species across 2 weeks in this area. It was a challenge to assemble all these different technologies, but also disciplines where we had lots of taxonomists, molecular biologists, bioinformaticians, divers, staff to run the boats, and so on. It was a great human experience. And we ran also taxonomic workshops to kind of share knowledge about different taxons, how to recognize them, how their lifestyle, how to sample tissues from them, and how to recognize them taxonomically. So tomorrow a whole expedition will go to Guadeloupe in the Caribbean here. Guadeloupe is made of several islands, and the largest island has already been well explored by the National History Museum, but the 3 smaller islands here have not. And so the expedition will focus on these 3 here, sampling marine specimen during 2 months and bring them to Paris. So just to say a few words about where we are at the moment. So this is a summary, a pie chart with all the origin of all the samples which we have collected so far, which amount to about 500 samples, mostly from the Brittany coast, because that's where we spend most time, dedicated really to sampling only for ATLASea, but also from other areas along the French coastline. We have sequenced in total 4 genomes released to the European Nucleotide Archive so far. The first year was really dedicated to setting up protocols, as I said, and testing all the pipelines. And now is the time to expand from there and start increasing the throughput. In numbers, exactly. So this is a table which summarizes from left to right the kind of progress sample goes through from the collection and arrival at acquisition, if you like, at Genoscope, wherever it was sampled from, and then short read data, long read data, then long read assembly, high C for assembling into chromosome scale scaffolds, a curation, and then finally the last column here is the release to a public repository. So as I said, we have 4 at the moment that are released, but many more are somewhere along the line in the process. All the data is available on a ATLASea data portal, where you can see where samples have been come from, the different statistics of how many samples are where in the pipeline. You can search using keywords such as the taxonomic name, also the status at which it is, and so on. And you can download sequences from there. So that will be all for ATLASea for now. Just to thank all the people that have helped put the project together and are currently running it with us. So I'll unshare and be happy to take questions if you have some.
Thank you very much. Thank you for keeping the time as well. That was great. I've seen at least one question in the chat. I was also thinking if, when you have a moment, if you could share the URL for that data portal via the chat, that would be great.
Yes, it looks like Lillian has shared the ATLASea main site, and at the top you have a link to the data portal.
Brilliant. She was one step ahead of me. So Elva has asked if you have a capacity development component in the Caribbean Sea.
Right. not sure I'm familiar with the term capacity development precisely, but I don't think we have, if I have a vague idea of what you mean by that, in the sense that, for example, the expedition that goes there tomorrow is really coordinated by the National History Museum in Paris. They're sending a group there, and they're working with local scientists and the Regional Office for Biodiversity. And we will also have establish training especially for young students in genomics and the study of biodiversity through genomics. But really, we're moving a whole operation locally with all the equipment coming from Paris for sampling liquid nitrogen, molecular biology, and so on and so on. Because the 3 islands which we're going to visit have really very basic infrastructure. I don't know if that answers your question. We don't have long-term development components established in any of the overseas territories, except New Caledonia, where obviously there is a university there and research laboratories, and we're working with them more on the long term.
Okay, thank you. I think that answers the question. There's another question about collecting samples in sparse islands, in particular French territories close to Madagascar. I think this is
so in the Indian Ocean, we will be working with La Réunion, and also there is another project similar to ATLASea called Bridges that is more focused on biodiversity linked to fishing, and not only in French territories, but we will work with them as well around Mayotte and La Réunion again. We are also aiming, but this is not the Indian Ocean, but going further south to near the Antarctic, where there is a small island called St. Paul and Amsterdam, where we might go as well, but not in the Eparse Island and not close to Madagascar, as far as I know.
Okay, thank you.
But these are all the expeditions that are funded and organized through ATLASea. We will, of course, also welcome suggestions from the community at large to suggest species for sequencing, or even to send samples for sequencing following our standard operating procedures. So these could come from anywhere, as long as they are in the French EEZ, of course.
Okay, so if there's a call for that, we'd be happy to share this through OBON.
Okay.
That would be great. If you don't mind answering the next question in the chat, I think, and you can answer it while Nick is giving his presentation.
Okay.
The question is from Nick Pade.
Yes, sure. So applied to the project is, so there are several calls that will, of course, all the calls for students will be open internationally. There's no restriction there. Applied to the 2 pilot projects, I don't think so. I think these will be, we haven't launched them yet, and I don't know the rules and regulation that ANR, the French agency that kind of is our correspondent for all things administrative, what are the rules there, but I think that it will be restricted to French researchers. And then for submitting species, they could come from anybody in the world, as long as the sample comes from the French EEZ. That is a restriction.
Okay, great. Thank you very much again. And we'll move on to Nick Pade, who will be presenting on the EMBRC project. And if you don't mind, Hugues, responding to some other questions in the chat, that would be great. But we can also possibly come back to some of these questions in the general discussion later.
Sure, I'll answer in the chat.
Thank you. Okay, so Nick, we can see your screen in presenter mode at the moment.
I don't know why my computer always insists on doing it this way around. Hopefully that will
That's
solve
fine.
the issue.
We can see that fine now. Thank you. And the floor is yours.
Okay, thank you very much, Sophie, and thank you for inviting me today to talk about our genomics observatory, EMOBON, and a project that we are about 1 year into called eDNA Aquaplan, which links intricately into what we're hoping to achieve with our observatory. I'll jump straight into my presentation. And so just to start with why we wanted to create a multinational biodiversity observation program in the first place. So the way we're looking at things in Europe, certainly, is that we have a very mature marine observation system and program. It's well structured, it's mature, it's got standards, and it's been running for probably 2 decades now, except for biology. So at least the biology is pretty rudimentary. It's certainly not at the same level. A lot of it is project-based today, which is of course not sustainable, and therefore also not really generating the sort of data that we want to have to make big decisions, long-term decisions, modeling and prediction about what's going on in biodiversity in the sea at the moment. Indeed, there's a general lack of rigor in implementing standard operating procedures. We're a little bit like butterflies with flowers. Whenever there's a new technology or procedure, we jump to that and then we change our protocols, meaning that it's extremely difficult to get really good long-term time series. Of course, there's still a lot of questions around marine biodiversity of where, when, why things are happening. And therefore, also having long-term time series is extremely important. So really what we wanted to do was to ensure that we could support some of these and fill some of these gaps through developing an observation program on marine biodiversity led by a research infrastructure, because we're not a university, we are a research infrastructure, and see if we could catalyze some change. So the objectives that we set for our observatory was to sort of act as an incubator for marine biological observation. Indeed, we have the same problem as everyone, which is how do we fund our observation in the long term? But at the very least, we could start to try and implement various things that have been suggested by the international genomics observatory community and take some of the sting out of implementation by providing shared protocol and linking to established observatories, creating a network already of people interested in developing such an observatory. We wanted to make sure that we had protocols and data management plans that were openly available so that anyone wanting to build an observatory would be able to access them and have something that they could build upon if they were starting from zero. In terms of the observatory that we're building here in Europe, we wanted to, well, not really take them over, because also we knew that we wouldn't be able to do that. And there are some observatories that have been going for a long time, but at the very least, we could add a component on genomics that would be shared amongst those observatories. And it would provide a baseline that was running across it and enable at least one level of comparability between them. Of course, we then also have to follow national and international regulations such as ABS, was access and benefit sharing, and national permits and so on. And we wanted also to take a crack at this in terms of could we facilitate it and could we handle that burden of reporting and permits for our partners. And then finally, we wanted to set this in a context of global collaboration. We didn't want this just to be something we were doing in Europe, but we wanted it to be a European contribution to having a better view of what's happening globally with the oceans. And so, as you can see on the right, we've started to develop some of these standard operating procedures. So we have a handbook of our different protocols, which are all available online. We've got guidelines for how to coordinate, how to deal with ABS, both for users and for genomics observatories and biological resource centers. Okay, so what we have today is the European Marine Omics Biological Observation Network. We have 20 participating observatories. We're spread across 10 countries. We're currently covering from the Arctic down to the subtropical in the Red Sea, and we cover a broad range of habitats throughout Europe. So I think we are definitely the biggest coordinated observatory in Europe, but probably also country-wise one of the biggest in the world today. So we're pleased about this, and think it's also quite an achievement to have shared protocols across multiple borders. Okay, so a closer look at our observatory is we have essentially three major habitats that we're looking at. We're looking at water column, we're looking at soft substrates, and we're looking at hard substrates. We have implemented mock communities for our sequencing. So in terms of having a positive control that we can run with all our sequences, we have that we've built from our culture collections. I should also mention that this is available if anybody wants to work with it. And we can also create these for you for particular areas if it's covered by our culture collections. In order to minimize error and user error in our observatory, we've also centralized all of the DNA extraction and sequencing. So just like Hugues, we work with the French National Sequencing Center, Genoscope, who's also been very, very helpful in terms of improving a lot of our standard operating procedures, extraction protocols, and so on. So they've really been a super helpful partner to work with. We're implementing full quality control on all of the data before it's being released. We have implemented quite high, what we consider to be quite high standards of metadata. So we're implementing full mixes and Darwin Core for our environmental data. So we should have very rich information accompanying all our sequence information. We've also managed to facilitate the ABS reporting. So rather than having to report on every single sample we take, we've now managed to negotiate with the countries that we report on everything once a year. So that's substantially reduced things. And this is so hopefully also a model that we can transpose to other places on how to facilitate what is a considerable administrative burden in terms of declaring the use of our sequences. We are at the moment still working on the data release. The data is all going to be open access and accessible through open access portals. The quality control, especially on the metadata, is taking quite a lot of time. As it turns out, there's not a lot of automated systems, so a lot of this is still being curated by hand while we develop the tools. But we are hoping that the first data publication will come before the end of the year. Indeed, we've chosen a data publication format so that we can provide citable reference for the technicians involved, and then that also gives 3 or 4 months of data in one go that people can then cite through publication, which is all that we ask for. Okay, so just a quick look on what we're actually doing with the protocols. So the water column is the only protocol that is obligatory in EMOBON. So all of our observatories are carrying out this one. It is microbial focused. It is a size fraction sampling, and we're doing full metagenomics on the water column samples. Because the reason we've taken a metagenomics approach is we wanted the data to be as broadly usable as possible. So not just barcodes that would allow for identification of species, but we would then also be able to start digging into what is happening at the level of genes and expression and so on, based on whatever we are seeing in the environment, whether that's heating or El Nino years or just a general human impact on what's going on. On the substrates, we got the microbial component, but we also have a myo and macrobenthos that we do every 2 to 3 times a year, where we are looking at the metabarcoding. The microbial component is still metagenomics. And finally, we also implement the ARMS protocol that's been developed by the Smithsonian. So these are long-term deployments of settlement panels. So essentially what the data this provides us with is what is settling in the environment. And this is a really good indicator, particularly for a large number of invasive species that we use it for as well. And there we are doing taxonomic identification from pictures, and we're doing metabarcodes as well. The whole setup that we've built is pretty simple in terms of it just uses filters and a peristatic pump. So it shouldn't have any major infrastructure requirements in terms of doing the sampling, which was an important part for us. So how does this work in terms of the data then? So we have all the sampling done locally. We then have all the data processed and done centrally at Genoscope. All our metadata ends up in OBIS and GBIF, and for those in Europe, it all runs through EMODnet as well, which is the European portal for data. And all the sequencing and raw sequence information will be available through ENA, so the European Nucleotide Archive. And so all of this will be publicly available. So where we are so far in terms of some achievements. So we have a position paper that came out last year explaining what the observatory is about and what we are hoping to achieve with it. We have all our protocols are available on our website, free to download, what we call the EMOBON handbook. We're also now working quite hard on the bioinformatics side of things, and I'll talk a bit more about MetaGoFlow later. So, but what's become very apparent to us is that, well, a lot of the tools that we want to use to work with this stuff, well, doesn't exist or can't handle the volumes of data. So this is another large piece of work that's ongoing. Well, we also consider that our endorsement by OBON to be quite an achievement. This was really important to us. And so we are really happy to participate in this network and the discussions that we're able to have with the like-minded people. Also to mention that in terms of also pushing the use of omics in marine observation, we've also been one of the funding members for an omics bond that really integrates the use of biomolecular techniques under the Biodiversity Observation Network of GeoBon. So moving on to the bioinformatics really quickly. So first of all, we are working hard to implement the IOC data recommendations on fair data, meaning that we are really doing everything we can to have data management plans that ensures that our data is fair, so mainly findable and interoperable and reusable by other people. We are working in general to develop more tools and pipelines for environmental genomics. So as soon as these are available, we make them publicly available and we publish them so people can use them. So really an insistence on open access tools that allow us to work with the same tools all over the place. So the two major developments that we have so far is MetaGoFlow, first of all. So we developed this tool to work specifically with shotgun sequencing data. Essentially, it assigns taxonomic identification to metagenomes. So rather than have to do this by hand and by individual, we can run this through an automated pipeline that does it for us, and that substantially reduces the amount of time it takes us to curate the data. So the data analysis is timely, it's standardized, and it's reproducible. So we also think it's a good contribution to general in science. And it's based on a tool that sits in the EMBL EBI network. So it's called Magnify. For those of you who are familiar with it, it basically is a tool that works for microorganisms. It's freely accessible anywhere in the world. So it's an open resource that is built upon that you can also use. The second tool is the part of the Tesseract that another infrastructure called LifeWatch is working on. So here we've been collaborating to create a pipeline for identifying invasive species from metabarcodes. It highlights any invasive species basically in a sample based on the worms database. So this is an ongoing process of improving our capabilities in Europe to deal with identifying, predicting, and managing invasive species, which might be interesting to a number of international audience as well. So, so this leads us on to also our new project, eDNA Aquaplan. So essentially what this project is about is to create a digital ecosystem for the next generation of aquatic biodiversity monitoring. By that, essentially, we mean linking reference libraries. So one of the major bottlenecks that we've identified in the use of our genomics observatories is really the limitation of reference material. So rather than trying to create one reference library to rule them all, which has been sort of a very common approach, we are aiming to see whether we can create a digital ecosystem of repositories that integrate reference libraries rather than collect everything in one place. So essentially provide them with the information of how to link them to one another. So we want to look at standardization and harmonization of data formats and metadata standards and ontologies, so we can really push the quality of the reference material up. And then essentially develop the underpinning infrastructure required for biomolecular observation by strengthening our access to reference material. There's a link to the website here and also to LinkedIn, so please follow us because we'll probably be making some requests and sharing the outcome of the project. Very briefly, just to say that so far we've been identifying who's active in the space, looking at what the current standards are that are available, paying particular attention to the UN Decade Data Strategy. And we're really keen that this isn't just a European initiative, but really we want to link as much as possible to reference libraries outside of Europe. So our recommendations will come by the end of 2026, but this is likely to be ongoing, and we'll keep updating everyone in OBON on where we are with the project as we go forward. And so just to move on to my final slide, just to say that yeah, we're really happy working in OBON, and we would really strongly encourage anyone who's in this type of work to join us as well. It's been a great forum for linking like-minded people. It's been great also to speak to people and really try to get to terms with whether we can achieve global observation, and I hope really pushing our ability to do that is something we're going to do more of. It's been a place for sharing resources, and I hope this will now also move towards including reference libraries. That we can really work on that area as well. So I'll stop there. Thank you very much again for having us today. I'm happy to take any questions, or if you have anything else, just email us and we will get back to you. Thank you very much.
Thank you, Nick, and thank you for the plug for OBON and for getting more initiatives involved, which of course we're very open to. There was a question, I think it was for you, what open access portals are you planning to publish the data on?
So all the data is going to be available through ENA, so essentially GenBank. So you go to GenBank, you'll be able to find the sequences when they're available. All of that will be included also in the data publications, and we will be putting a direct link to the data archives within ENA, because everything's going to be published as a project. Everything will be findable and accessible in there. The whole idea with the project is to make the data available to anyone and everyone who wants to use it. The only requirement is that we ask you to cite the data papers and acknowledge us. That's all we ask for. So yeah, that's how it should be.
Okay, thank you very much. Let's see any. Oh, another question. Are you planning to submit the processed biodiversity data to GBIF OBIS as well?
The actual, what we are not planning any major data products at the moment, because, well, we've kind of shied away from that and let people do with the data that they want. But any product that we do decide to make that goes beyond just the quality controlled sequences, yes, the aim is to make it available through portals as like those. That's, I mean, a general mantra of this project is to make everything available to anyone who wants to use it. So it's an example and then a resource for people working on marine microbes and interested in sort of a larger scale microbial ecology.
Thank you. And yeah, just to add to that, as a general concept within OBON, we are trying to encourage OBON projects to submit data to OBIS, and we're working very closely with the OBIS IOD Secretariat, and we'll be arranging some kind of demo / training for OBON projects to learn a bit more how to go about submitting data to OBIS. So once again, thank you, Nick. I think we'll move on to Kristen. I don't want to run out of time. So and then again, we hopefully will come back to some discussion at the end. Okay, so Kristen, I can see your presentation.
Wonderful, thank you. Thank you, Sophie, and thank you to OBON for inviting me to speak today. I'm here to present a project that was recently endorsed by the UN Decade of the Ocean, the Canadian Marine DNA Library, or CANDLE, and MPA eDNA monitoring. So this is a two-part project. So my name is Kristen Westfall. I represent the Department of Fisheries and Oceans in Canada. I'm located in the Pacific side at the Pacific Biological Station. And Emily Rubich, who's on the call today, she is the co-lead on this project. And Emily is also located on the Pacific side of Canada, but her home institution is the Institute of Ocean Sciences. So an overview of my talk today, I'm just going to briefly introduce the project, give a brief overview, and go talk about the partners involved, and also the background, so the reasons why are we doing this in the first place. And the bulk of my talk will be focused on the objectives, so progress of what we've progressed so far and our future work. So this is a project that started in 2022, so we have progress to report as well. Okay, so biodiversity monitoring is a core tenet of conservation management, and Canada has established a series of guidelines for MPAs that emphasize the need for non-destructive multi-species survey methods. And eDNA metabarcoding is revolutionizing the field of biomonitoring. And this project has initiated eDNA biomonitoring in marine protected areas and areas of interest in Canada's Pacific and Atlantic Ocean regions in order to collect baseline biodiversity data on species diversity and distributions. We're also creating a high quality and accessible reference DNA sequences for which we know are critical for the effectiveness of eDNA as a biomonitoring tool. So the Canadian Marine Biodiversity Library is an expertly curated and high quality DNA reference sequence database that we are creating in order to support the uptake of eDNA results for science-based advice within Department of Fisheries notions and beyond. So pairing our reference library with the MPA biomonitoring surveys, this accelerates eDNA operationalization across Canada. So the partners in this project, I mentioned Emily is the co-lead in the Pacific region. We also, this is a national scoped project. So we have collaborators at the Bedford Institute of Oceanography in Nova Scotia, Ryan Stanley and Nick Jeffrey. And we have some external partners with the Matt LeMay is with the Hakai Institute, part of the Tula Foundation, and Jennifer Sunday is with McGill. And we have also some affiliations with some institutions that support the reference library generation and the Royal Ontario and Royal BC museums. They receive vouchered specimens for their collections, and Illumina also is supporting us with our sequencing efforts. So Canadian territorial waters consist of 3 oceans, the Pacific, Arctic, and Atlantic Oceans, and multiple seas. Canada's EEZ is the 7th largest in the world at 5.6 million square kilometers, and we've committed to protecting 30% of this by 2030. And marine conservation areas in Canada have increased by almost 900% in the last 5 years. So we now sit at about 800,000 square kilometers under some form of protection. But Canadian MPAs are large. The mean size is about 1,000 square kilometers, sorry. And they're also quite deep in general. So 75% of MPAs are deeper, have habitats that are deeper than 500 meters. And they're also isolated. So about 45% are more than 10 kilometers from shore. And as you can imagine, monitoring these isolated, deep, and large open areas can be quite challenging. So most marine protected areas are also data deficient. And as I mentioned before, Canada has passed recommendations against the use of bottom contact gear for monitoring marine protected areas, which makes eDNA one of the best tools that we've identified so far. And we also know that based on biodiversity studies at scale in these large, deep, and remote areas, using traditional methods, these surveys can take many, many years, decades, and they're also extremely costly. So we also are looking for an efficient biodiversity tool. We need to detect species across a broad range of taxonomic groups. And MPA monitoring strategies and protocols are currently being developed at the policy level because we are rapidly expanding the number of marine protected areas in Canada. And so we are trying to officially develop monitoring strategies at this point. So the first main objective of this project is developing the Canadian Marine DNA Library, and this is also known as CAMDL. It's a DNA reference library of mitochondrial genomes from vouchered specimens, and a web repository containing the genetic data has been developed and will be online this fall. So the reference library adheres to 3 pillars that we developed for building better reference libraries. At the top, we have complete. This refers to complete mitochondrial genomes and nuclear ribosomal DNA, and we obtain this through genome skimming techniques. Quality assured refers to vouchered specimens that are accessioned in museum collection. So this enables defensibility of eDNA results, especially coming from a government regulatory setting. We need to, at times, be able to defend eDNA results in a court of law. And accessibility, the final pillar, this refers to the web platform that we developed. This is a user-friendly platform and enables people to create custom reference libraries for a range of eDNA applications to suit specific needs. So the bottom image, because the website isn't live yet, I was going to just share a few screenshots. The bottom image is a screenshot of the homepage for the repository. So the heart is the CAMDL database, and this contains all genes from the mitogenomes with standardized biodiversity metadata terms aligned with GBIF and OBIS, and standardized genetic data terms aligned with MIXIS. So users can generate custom reference libraries with only their taxa or genes of interest by searching the database with several terms. So this next screenshot is actually the search result page. On the left, you can refine the search results. So you can keep refining to get exactly what you want. And the criteria on the left are the same criteria as in the main search page. And on the right, we have the results. So this is test data, so the names are all the same. But the user can hover over each record and get the full name of the record. You can select the records. You can select all of the records at once. And then at the bottom, you can download a FASTA formatted data file, genetic data file. And then you can also download a separate CSV formatted metadata file for the same records. So CAMDL is just finishing up its pilot phase, which went from April 2023, and we have about 6 months left till next year. But the main outcomes for this pilot phase were to generate this web platform and populate the database with 300 mitogenomes. So how does CAMDL fit within the wider scope of MPA biomonitoring? Well, we're incorporating this reference library into a case use framework for DNA-based biodiversity monitoring in general in Canada. This is important because the case use framework will be used to increase the uptake and operationalization of eDNA tools on a larger scale. And as an eDNA scientist in a large government organization, part of my job, sometimes a lot of my job, is to translate the confidence of eDNA results to managers and policymakers. And so one of the case use frameworks that we're developing is for a newly implemented MPA, the Tengwang Hatchahuaycoxigas MPA, or otherwise known as the THT MPA. This is one of Canada's, Pacific Canada's largest at 133,000 square kilometers. It has an incredibly diverse marine wildlife owing to the network of hydrothermal vents and seamounts on the ocean floor. So half the mitogenomes in CAMDL are from deep sea invertebrates collected during the 2023 Northeast Pacific Deep Sea Expedition. And on the right, we have a map outlining this is the boundary of the THT reserve. And these are some of the sites that reference sequences were collected from. Not all of them, because there's some seamounts here that we also had reference samples collected from. And by combining the reference DNA data with our eDNA results collected from the same areas, we're able to develop this use case framework for how we collect and report on DNA-based biodiversity data in general. So the second year of CAMDL, which was this year, focused on different ecologically and biologically significant areas in both the Pacific and Atlantic regions of Canada. So we collected specimens from major marine habitats like glass sponge reefs, high latitude coral reefs, nearshore environments like eelgrass beds. We also had taxonomic specific collections for rockfish, for example, in the Pacific region, we have areas for rockfish conservation. And so that's also a true target group for CANDLE. So the second main objective of this project is the MPA eDNA biomonitoring program. The first phase of this program is coming to a close next year, where we conducted a rapid biodiversity assessment to generate biodiversity baselines in existing or proposed MPAs. We're using eDNA metabarcoding for fish and invertebrates across multiple depths at each site, and three metabarcode markers. That's 2 for fish and CO1 for invertebrates. So over 3 field seasons, we've collected more than 1,300 samples from about 200 sites in proposed and existing MPAs. And most of these were sampled at 2 or 3 depths. So on the right, we have a map of the coast of British Columbia here. And this contains only the 2022 and 2023 sites. And this green area here, this is the THT Marine Reserve from the previous map. This is now an official marine protected area, just as of a couple of months ago. So we have collected eDNA from the surface all the way down to almost 3 kilometers, so a multitude of habitat types. And just in the 2022 data, so just from sites here that are highlighted in blue, we detected over 220 fish and 1,300 invertebrate taxa. So during the next phase of the project, we're going to establish the long-term portion of the biomonitoring program. We're going to research indicator species and incorporate an inside-outside MPA sample design to be able to report on conservation effectiveness. So as part of building this baseline over the last few years, we've also alongside build our technical and infrastructure capacity. So this includes everything from sampling, lab, and analytical workflows. We've been employing standardized survey design and protocols for different habitat types. The way we filter has also evolved, and now we're acquiring an automated filter, so we'll be developing additional sampling protocols for that. We developed a repeatable and scalable laboratory workflow based on Tag Study, which is out of Carolyn Bowman's labs. We've adapted this protocol to accommodate up to 1,800 amplicon targets. And then we're also using the NovaSeq platform to do deep sequencing on these amplicons. The project this year, the project is developing new bioinformatic protocols that meet FAIR and CARE principles, specifically targeted for NovaSeq data. And so these have already been developed, and we're going to be automating these bioinformatic protocols. And in the future, we're also going to be developing specific data handling best practices. So this is things like looking at how we deal with blanks, how we apply minimum read numbers, minimum replicate numbers, et cetera, because we know that one size doesn't fit all when it comes to the downstream analytical applications. So we're looking at defining best practices for this. So our final objective is really just tying it all together, getting to the question of how are we planning to use the reference library and the eDNA biomonitoring data in the future to increase the use of ocean knowledge. So this isn't a straightforward answer, as data use will be somewhat dependent on the translation or uptake at higher levels of management, which can be hard to predict in some cases. But I already talked about the use case framework for the reference library and the eDNA data from the THT MPA. With the development of ongoing monitoring, indicator species, inside-outside MPA survey design, we also plan to report on the effectiveness of conservation measures. And the presence-absence data from the baseline assessments is being incorporated into species distribution models, eventually also for marine spatial planning. We are the baseline data is also available to assess changes in species distribution over time, either due to climate change or changes in human use, for example. And the species at risk detections that we get will be available to be integrated into recovery and conservation planning for species at risk. And lastly, the best practices and guidelines for generating DNA-based observation data enables the data that is collected to be comparable through space and time. So the goal is to create a long-term monitoring project on the national scale that uses best practices and guidelines. And with that, I'd like to put up this acknowledgement slide and also take any questions.
Thank you very much, Kristen. I see that there's a few questions already in the chat. The first one was what markers are you using for the fish, MyFish12S, and which other or others?
Right. So yeah, so we are using the MyFish12S, a modified MyFish12S, to reduce the mammalian amplifications. And then we also use 16S region, marker developed for the 16S region. And those two are for fish, and then we use CO1 primer for invertebrates.
Okay.
And I see another question about protocols that meet fair care principles. So the findable, accessible, interoperable, and repeatable, the bioinformatics protocols, this is one of the areas I think in eDNA metabarcoding that hasn't been given as much attention as others, because bioinformatics it's a bit of a black box for a lot of people, and there aren't a lot of automated protocols, bioinformatic protocols out there. And then when you start to change the sequencing platforms, you say you jump from the MiSeq to a NextSeq or a NovaSeq, so those same bioinformatic pipelines will not work, or they won't work in the same way. So when I talk about developing bioinformatic protocols, we are specifically targeting NovaSeq data, large scale, terabyte scale NovaSeq data. And when I mean automated, will enable the results to be repeatable, because you can have versioned pipelines using automated software. And how are we implementing this? Well, we haven't implemented it yet, so we're still in the process of deciding on how to do that. We don't quite know yet, but everything will be published and made publicly available. And I see a follow-up question, evaluating the impact of policies and how have you defined success? So what does that refer to? I just wanted to clarify.
So you mentioned that you were going to use the eDNA to evaluate the impact of some of your marine protected areas. And so something we are looking and discussing a lot in Europe is, well, what's the baseline? How do we evaluate policies, and how do we define what is a success? In other words, was the policy effective? Have we improved biodiversity? But that can mean a lot of things. It depends what you're looking at. So that's what I mean by defined success. How, in terms of your monitoring and the data you have, when you look at that data, what has you in Canada decided means success? And did policy work, and it was worthwhile? Right.
Yeah, so I think we're probably at a similar stage as you, is defining success can mean a multitude of things, and there isn't just one thing, and how also do you measure success as well? So looking at eDNA, it's difficult to do quantitative measurements. So obviously looking at increase in biomass is not as straightforward with metabarcoding. However, we are looking at doing quantitative PCR for indicator species as one option of looking at potential for success, or just like looking at the effective conservation measures. So, and employing inside outside MPA survey design, and also I think this also there'll be an element of time involved here, in that looking at this isn't something that we can measure in 1 year. It'll be decades of measurement. So that's where our long-term biomonitoring program really comes into play, defining those sites where we're going to be looking at long-term. So that's a great question, and a very difficult one to answer.
I was hoping you were going to give me the answer. Thank you very much, Kristen.
You're welcome.
So Jennifer Sunday has got her hand up, probably to expand a bit on her comment in the chat. Yeah, Jennifer?
Yeah, can you hear me okay?
Yes.
I was planning just to listen, but Nick, I think that's a really great question. And Kristen referred to a broader project that we're working on with Emily and Matt and through a genome funded project. I could talk about it on Monday, but it's specifically to look at, I mean, next week when I'm speaking on a power analysis for biodiversity data from eDNA. But as we learned, it was like actually general, like we don't really have power analysis for biodiversity change. And so we're talking about just basic species richness or species turnover metrics. How do we know that how much sampling do you need to know that you've detected change? And given your amount of sampling, what's your likelihood of detecting change if it was there? So that might be interesting. It's kind of nice to know that you're talking about it too.
Thanks, Jen. There's another question in the chat. Is eDNA analysis essential to redefine the limits of existing MPAs and or to establish new ones? Can it be used alone or in combination with other methods? Which is a great question, I think. Kristen? But you're muted.
Yeah, sorry. Yeah, that is a great question, and I don't think that we would have an answer to that yet. We've really just started to look at baseline biodiversity data within our MPAs. We're sort of coming to the third year of this. We would like to continue to do baseline biodiversity analyses, but we're going to be establishing long-term monitoring sites in the second phase of the project. But this is a great question and something that we will hopefully be revisiting throughout the project.
And it's certainly something that OBON is interested in. And as I mentioned at the beginning, we're currently working on a workshop or preparing a workshop on eDNA applications for fisheries management. But the plan is to have a workshop after that on eDNA for MPA management, which will kind of bring together different stakeholders to look at that question and to kind of bring in scientists who can help to manage expectations of policymakers who might be interested in eDNA for MPA management. Jennifer, did you want to follow up on Emily's comment or vice versa? No, she's lowered her hand. I just wanted to highlight, I put it in the chat, but when you were mentioning protocols and sharing protocols and all that, I don't know if you just share them as kind of PDFs or publications, but OBON is fostering this system for standardized sharing of protocols, which Katie Pitts is leading from EMBRC. She was leading a project within OBON, which has now officially ended as a project, but it's transitioning to being a working group within OBON, with the same objective of facilitating sharing of protocols and also protocols for different stages of the process. So as you mentioned, bioinformatics is something that she's quite interested in getting protocols for in the system, and also things like permitting and kind of other things that other than the actual sampling or analysis or sequencing kind of steps. Okay, so we have about 10 minutes left, and I'd like to use that time to have get a bit of a feel for how all of your projects think that they could work together or work more effectively within OBON? You know, what can OBON do to support or add value to what you're already doing in your regions or in your countries? So I would like to kind of open that up to the speakers, but I know there are representatives of other projects here, so if anyone wants to chime in, please feel free. Kristen, ladies first, do you have any thoughts on, you know, what would you like to get out of OBON as, you know, this network of projects and different groups working on similar things? Is there anything you think that OBON could help with? The community
being new to OBON, I'm really just sort of getting a feel for the people and the projects involved. But one of the main advantages that I see now is joining a community of practice for biomolecular observations in the ocean. And as Nick alluded to it in his presentation, can we reach a global observatory? Can we reach status of a global observatory? And I think the best way to do that is to get everybody in the same room, so to speak, same virtual room, and get the conversations flowing and just getting everybody together. So I really, as far as I can see now, I think that's one of the great advantages of joining OBON. And I'm really looking forward to learning more about it as we go along.
And we'll be in the same actual room in a couple of months, I think, in Plymouth. So that's always great to have those live in-person conversations as well. Nick, going in kind of back in reverse order.
Yeah, I think so. I think Kristen has already said it very well, but I think there's three things that I really would like to see happen in OBON. And I mean, the first one is kind of around Kristen's point, which is it'd be great at some point just to sit us all down and start throwing all the data on the table and see what are we seeing. You know, even just find out, are we even comparable at the moment? Can we do something? That would just be, I think, an amazing achievement. The only people who've really done this sort of on a big scale in a very visible way has been Tara. And Tara's been basing this on like one sampling site or only just a few. And here we're talking time series, you know, so we are already able to probably look at a couple of years. And can we look at, you know, El Nino years and whatever else, and just start to get a snapshot, I think would be super exciting. That's the first thing. The second thing is around the capacity enhancement. I would love to also start seeing more people coming to the meetings from outside of Europe and North America, and Australia as well, because we get good colleagues from there too. But there's a plethora of other places that, and so we've seen in Latin America, for example, some really cool stuff going on, and we're just not managing to get to people and meet up with them. And I think that's, for me, a real shame. It would be amazing if on the back of OBON, we could really start engaging with those regions, going there, learning from them, and sharing what we've figured out as well, because I think all of our information's been really hard won. And the third one is sort of just try and agree, not necessarily on priorities, but just to say, okay, well, what are we all working on and what are we stuck on? We're likely all working in 3 or 5 places on the same problem. And so putting our heads together, especially between regions, I think is going to be really exciting, because we're, especially in Europe, we have our bubble of how we run projects and look at it and so on. And it's always really nice to hear other perspectives than outside of our bubble and how people are approaching it and the science that they see and what they have available that we don't know about. So sort of agreeing on working on a number of challenges, which are probably likely going to be bioinformatic, I think would also be really exciting. So it is a small community. So can we share the resources that we have to really amplify what we're doing and collaborate much more strongly across borders and continents than we are today?
Thanks, Nick. And I think we're getting there with the, you know, reaching out to different regions. Unfortunately, this particular time is not good for our Asian colleagues. We had to kind of schedule it to be convenient for the speakers, but we're certainly starting to reach out to Latin America and Asia, and hopefully Africa as we go along. Hugues, as one of our newest projects with fresh ideas,
Well,
what are
I
your
don't know about
thoughts
fresh
on
ideas, but
OBON?
as I said in at the beginning of my talk, we're a bit like outliers to the OBON community because we're not really in the observation trade, if you like. And so I think what OBON could bring, or the OBON community could bring, is because you see, I think ATLASea is really like other reference genome project, is at the other end of the spectrum compared to projects like well, that are in the using eDNA or barcoding technologies to monitor biodiversity. So I think reference genomes can really help those projects, especially when you're sequencing samples where you don't know what the species are really. You see a fragment of DNA, but you don't know exactly what the species are. And so at the other end of the spectrum, we're sequencing one genome per species. We're having a photograph of the species. We know exactly taxonomically which one it is. And so I think we need to find some way to meet across this spectrum. And it'll take some time because I think we need a critical mass of reference genomes for it to be really useful for eDNA studies. And we're really still far from being really practically useful, but eventually I think we will. And so probably, for example, for ATLASea and biomonitoring programs along the European coast, I think that could be effective in 3 or 4 years, for example. So in this time lapse, I think yeah, it will be nice to anticipate and work with, as Nicolas said, developing bioinformatic pipeline to start processing that interoperability of databases that collect eDNA on the one hand and reference genomes on the other hand. Yeah, starting to think about that and see how we can meet halfway in the spectrum, going from eDNA to reference genome is what I'd like to discuss.
Okay, thanks very much. So unfortunately, we've come to the end of our session. So I'd just like to thank all the speakers once again for the great presentations. I certainly learned a lot from them. And I'm very excited to have all these new OBON projects involved in our network. So we will have our next webinar on Thursday at the same time? No. Not the same time? Maybe earlier, 2 p.m. So the next one will be bringing together projects working on methodologies. The Bebop project, which I mentioned earlier on the protocol sharing, will give a presentation. There will be one on making eDNA fair, as fair data is something that came up, that always comes up, and a project, another project from Canada on optimal timing for eDNA. So it will be great to see many of you for that one, and it will be at 2 p.m. UK time on Thursday, I believe. But please check the website and the registration link, which will give you the time in your own time zone. Okay, so thank you to everyone who joined. It was a great turnout, and thank you once again to the speakers. And hopefully see you next time.
Thank you. Bye-bye.